Hey, it's Joel Duff. Welcome back to my channel. Today, a quick episode. Well, is anything ever quick when I say it's going to be quick? We'll see. An episode, I'll just call it an episode of Critiquing Creationism, wherein we are going to look at a series of Facebook posts that have been showing up on a particular Facebook, well, actually multiple different Facebook groups recently. And these posts are raising a question, or no, they're really raising a challenge to non-young Earth creationists about the percent similarity of DNA of one organism to another, saying that this is completely bogus. And actually, the percentage similarity actually somehow proves that the world can't be young and evolution hasn't happened. Okay, that's a really big claim. Now, maybe you've heard people say like, bananas are 50% similar to human DNA, or that some animals are less than 50% similar at their DNA level to human beings, or some are 90% similar, or chimpanzees maybe 98% similar, but some other young Earth creationists claim it's only 80% similar. What's up with all these percentages, and what do they mean, and what do they tell us? And is there any merit to the argument that I'm going to show you from these Facebook posts? Is this actually proof that evolution in billions of years are false and that God must have created things? Well, the implication is God instantaneously created these things. So here's an example. This is actually from today. Why the similarity in DNA between kangaroos and mankind proves evolution in billions of years are false and God created all things. And it goes on to state, supposedly there's a 50% similarity in DNA between kangaroos and mankind. So only a 50% difference, right? The supposed last common ancestor of kangaroos and mankind lived 150 million years ago. I don't know. I didn't check that piece of data, but it sounds reasonable, right? Based as in terms of the conventional thinking here. So that means 150 million years ago is how long you'd have to go into the past to find a common organism, all right, which gave rise to the lineages that lead to kangaroos and human beings. Now, he goes on to say, assume there's a 2-year average intergenerational time for the entire line leading from mankind to from their last common ancestor. So that would be 75 million generations, right? 75 times 2, 150 million years. Not sure where the 2-year average is, since it takes a whole lot. I mean, I don't know any 2-year-olds that are giving birth to, offspring. I don't, possibly in kangaroos, that could be the case, but certainly not in human beings. But this is a minor quibble. We're going to talk about this argument a little bit more, but let me show you some other examples. I just want to show you how social media is being bombarded with this particular claim right now. So for example, this is just in the space of the last week or so. Why the similarity of DNA between platypuses and mankind proves evolution in billions of years are false. Supposedly there's a 60% similarity of DNA between platypuses and mankind, so therefore only a 40% difference. Suppose the common ancestor lived 160 million years ago, and then the same language. Oh, and then what about this? Supposedly there's the similarity between birds and mankind proves evolution, but there's only a 60% similarity in the DNA of birds and mankind, so therefore only a 40% difference. The last common ancestor was some 300 million years ago. And what's the similarity between zebrafish and mankind? Or what's the similarity between green and old lizards and mankind? They're only supposedly 30% similar. How can a green and old lizard? You might be looking at this and saying to yourself, "Wait a second, shouldn't lizards be more similar to human beings, being that they're land vertebrates, versus a fish?" All right, which in even I think if you don't know much about evolutionary theory, you probably know the suggestion that fish came before land animals or land vertebrates, and therefore should be older and share a common ancestor farther back in time. So you might think they should be more dissimilar based on their DNA sequences than, say, a lizard. So here you have 60% for a fish compared to humans, and only 30% similar for the old lizard. Where do these numbers come from? Are these numbers accurate? Well, we'll discuss that in a few minutes. A couple more. Again, this is just it's sort of the same thing regurgitated over and over and over again, the same kind of argument. Why is the similarity between sea urchins and mankind? How does that prove that evolution in billions of years are false? Suppose there's a 70% similarity between DNA of sea urchins and mankind. Well, sea urchins are much more distantly related to land vertebrates like human beings, even compared to fish, and yet he's saying that there's a 70% similarity between those. Now, I didn't check like well, there are no references. All right, so there's no references provided here for this data that I remember. We'll look at one of these, and so maybe I'm wrong. But off the top of my head, these numbers don't sound quite right to me, but I'm going to have a lot of caveats as to the difficulty of determining what is sequence similarity. What does it mean that something is 70% similar? That's not a trivial thing to determine. And lastly, what about bananas and people? 50% similarity. Well, those are plants, not even an animal. So a plant and an animal being 50% similar. Okay, what's going on here? First, before I show you some reasons why similarities can be very deceptive, similarity statistics can be very deceptive, let's read the entire post. I'm just reading you the top portion of it, but this is actually a fairly long Facebook post that gives the rationale or the argument, actually the calculations for this. So let's run through that as quickly as I can. And then we'll just talk about a few things that need to be considered and why this is really no proof, all right, that evolution in billions of years are false. I'm on a public Facebook groups. I think I'm okay sharing this information. This particular author, it doesn't have I don't know anything about their expertise. I can't find out anything about them. So I don't know whether they have any scientific knowledge at all. Let's just say they're a prolific poster of copying creation information and sharing it. But this seems to be a slightly more novel argument. I haven't really seen this exact argument in the young earth creationist literature, so I think this is something that he's coming up with himself. So let's take a closer look at this. Suppose that there's a 50% similarity. We'll just stick with the kangaroo example. 50% similarity in DNA between kangaroos and mankind, so only a 50% difference. Suppose the last common ancestor lived 150 million years ago. Assume a 2-year average intergenerational time. Again, he's going to use 2 years for all these particular examples. And one of the problems of doing that is of course, generation time is very much tied to the number of total mutations that are going to happen. Because if you are a short-lived organism, you're going to have a lot of mutations because you're going to have a lot of generations, even within 100 years, right? If you're a housefly, right, and you're making tens of thousands of houseflies, and you're doing that a couple times a year, right, you're going to generate an enormous number of mutations. So keep that in the back of your head as we look at his example. But if you do some simple math here, if it was every 2 generations, every 2 years you have a new generation, and that passes on 60 mutations per generation. I'm sure he gets that number from sort of the basic literature in terms of like human beings. And of course, are human beings exactly like bananas and exactly like kangaroos and exactly like fish in terms of the number of mutations they pass on to their offspring? No, but he's using that same number for all different things. I don't think it makes a significant difference. This isn't a huge problem, but it would be inaccurate to use the 60 across these different organisms. But nonetheless, for you and I, right, I have kids. My children have probably somewhere between, they started out at least with 60 to 100 new variations, right? New mutations in their DNA that my wife and I don't have, right? Well, that were made in the sperm and the egg. And so those are new contributions to the next generation. And so this is on average sort of the number of new mutants that are formed every single generation in the human race. So he's just using that rough number and saying, well, if there's been 75 million generations from the last common ancestor 150 million years ago, and you just take that forward 75 million times 60 mutations every generation, you would have had along the lineage leading from the common ancestor to us 4.5 billion mutations, right? 4.5 billion mutations, right? That is, is pretty accurate here. That's about 150% of the genome size of human beings, right? If we have a genome of 3.33 to 3.3 billion base pairs, 4.5 billion base pairs, right, would be more mutations than the entirety of one copy of our genome. Now, the thing is, again, there's a little inaccuracy here as well. We have two copies of our genome in us, right? And so when we say that we've inherited 60 new mutations, that's 60 mutations over both of our copies of our genome, because we got one copy of our genome from mom, one copy from dad. In reality, we have over 6 billion base pairs in our genome. And so 4.5 billion isn't actually even all of the human genome, but I'm going to grant this person this particular idea because the point he's trying to make, and I think the thing that will surprise readers who are oblivious to mutations and substitution rates and how this happens, right? And what genomes look like, is they're going to hear this. They're going to say, like, wait a second, I've got 3 billion base pairs of DNA, but over that much time, there's been 4.5 billion changes. Well, that seems like then my entire genome has been changed out. If my entire genome has been changed out, and oh yeah, look what happened here. You also have to consider these are two lineages, right? You started with a common ancestor. That common ancestor then had 75 million generations leading to us. Really much less because, of course, we're not reproducing every 2 years. Let's say it's every 10 years, right? So that's 5 times fewer lineages than that. But let's keep it simple, 75 million generations, and 75 million generations then from the last common ancestor to kangaroos. And if you assume they have the same number of mutations to their genomes every generation from that common ancestor, that would be another 4.5 million mutations along that lineage. And so from the common ancestor to those descendants, there have been more mutations than the totality of the number of actual positions in a genome altogether. Right. I just said that was actually wrong, but well, let's just go with like the genome is 3 billion base pairs and you have 4.5 billion base pairs. How could that be? It would seem like then kangaroos and human beings they can't have anything in common. Can they have any sequence in common if they've both completely, utterly changed out their genome from the last common ancestor and had more mutations than there are sites? There shouldn't be any similarity at all between them, right? The calculation shows that, let me see here. That's about 1.5. That means each base pair in the genome of kangaroos would have changed on average 1.5 times. Every single base pair. Now, if you have, again, let's think about this. Again, a simple example. If I told you there were 3 billion changes that had occurred to the genome over some period of time, and your genome size is 3 billion base pairs, that would mean on average, each site has changed, right? One time. But you know that that's not actually what would have happened. I mean, some sites would have changed many times and some other sites might not have changed at all. So keep that in mind too. That's an average. We're going to see that there's a whole lot of other factors, which means that average is much more lumpy than it might sound. Yeah, lumpy. I know I got to show it to you. I'll show you in a minute. So if you combine the two, it comes to 300% being between kangaroos and mankind. I guess he's saying there's a 300% change from the last common ancestor to both of those lineages. I don't know if that's really the right way to talk about that, but again, let's grant that that sounds pretty like, whoa. I mean, 300% change. If there's been a 300% change, how could we be 50% similar? We should be 0% similar. That's what I think that the lay reader of this is supposed to get out of it. And I don't know if the author actually thinks that as well, that 50%, that's impossible. It should be 0% because every single base pair has changed. Calculations show that the last common ancestor of kangaroos and mankind can only be at most 25 million years ago if evolution is true. That is what he's thinking. He's thinking, actually, in 25 million years, if you have 60 bases change, right, in every one of those generations in those two lineages, that would add up to the entire genome in 25 million years. So in 25 million years, you should create an entire difference between those two different organisms. They shouldn't have any similarity at all. And therefore they can't be more than 25 million years old, right? If they have any similarity at all. And yet here these evolutionists are saying that they're 150 million years ago, they have a common ancestor. So evolution is false, right? Thus, evolution is false. Please note that proof refutes two major branch offs in the supposed evolutionary descent tree, from ancient mammal to kangaroos and from the start of the ancient mammal to mankind. Suppose there's 50% similarities in the DNA of kangaroos and mankind. So suppose only a 50% difference. So where did all the mutations go for both lines? And I think that if you've never thought about this, that's a completely and totally appropriate question to ask. You should be asking that question. Where did all those mutations go? Because I'm not going to deny the mutations didn't happen. Right? There are going to be 50, 60, 100 mutations every single generation to a genome that has billions of base pairs. It's just chemistry. I mean, you can't perfectly copy everything. You're going to make mistakes. It's actually amazing the mistake level is that low. We have lots of error correction mechanisms, all kinds of things we do to try to keep our error rate really low. And that is extraordinarily low, right? 60 out of 3 billion. It's incredible. And yet 60 is still a lot when you consider that 60 times millions of generations is a whole lot of mutations. Now think about it this way, though. How many human beings are on Earth, right? So if there's that many human beings on Earth, right, there's billions, right? And let's just say for the last couple, 100 years, how many new individuals have been born? Billions. If there's been billions born, then there's billions times 60, all right? There's hundreds of billions, all right? Hundreds of billions of new mutations in the human population, which is many, many, many times over the total size of the human genome. Now, of course, that's lots of different lineages. Not every lineage is going to have 100 billion mutations. But nonetheless, the idea is there's a whole lot of mutations. And that can sound kind of scary if you just spread, that many mutations across the human population. You'd be like, well, what's going on here? I mean, there's just a huge pile of mutations piling up. Do we have more mutations in us today than somebody living 4,000 years ago? It sure seems like it, right? Aren't we always adding 60? If we just keep adding 60 every generation, I could go back, and this is what this person is suggesting, right? This is their argument. If I go back 20 generations, that person should have had 1,200 fewer mutations than I have. Right. And if I go 20 generations of fewer, that's going to be 1,200 more mutations in my descendants 20 generations from now. And so the mutations are being more and more and more and more and more. Shouldn't they just completely take us over and completely wipe us out? Right. Or completely change us into something else? Now, I've done a couple other videos on this. So I'll try to remember to link those below because I don't want to go through every detail of those. Like I have a video on trillions of mutations, because in your own body, at my age, I've experienced quadrillions of mutations. Quadrillions of mutations have happened to me, right? I have trillions of cells, and your cell is just like reproducing, right? If I reproduce and have offspring, I'm going to give them 30 or 40 new mutations when I copy my genome. What are you doing in your body right now? You're copying your genome, all right? Millions of times. You're creating hundreds of millions of new mutations every day, right? So you have enormous numbers of new mutations in your somatic cells. And I don't have time to explain why it is that you're not going to pass all those down to your next of kin, right? To your offspring. But point is, lots of mutations happen. And it's easy to think that they must be accumulating, and therefore, how could two things be similar to each other millions of years later? They should have no similarity at all. That similarity should be obliterated by the mutations. All right, let's keep on going here. Before I come back and try to explain, like, how is this possible? How is it possible you're alive, with that many mutations? And how is it possible that you could go back, that you could have 50% similarity between an organism that's 100 million years old, between its last common ancestor? If you look at it, you'll see, he's pointing to a phylogenetic tree. He's talking about how many millions of years old it is. The accumulation of DNA mutation errors in nuclear DNA, especially in mitochondrial DNA in all species and all creatures that ever lived, proves that all species are less than 10,000 years. Less than 10,000 years old. The reason is the genomes of all species would have become too corrupted if species were too old. These facts refute evolution and billions of years completely. They also prove God created all things recently. So all fossils are less than 10,000 years old. All right, so we're saying, okay, I've defeated evolution. I've defeated billions of years. Life can't be more than 10,000 years old. And only explanation, now this doesn't actually prove that God did that, but we'll grant him that. So then all fossils must be less than 10,000 years old, okay, because there couldn't have been organisms more than 10,000 years ago. So yeah, so it's one thing after another, right? So this to him is like, this is the gotcha. I gotcha. Look at that. You're saying that 60 mutations occur every generation, and all I have to do is just use simple math, and boom, you're destroyed. I've completely refuted billions of years. I really should say millions of years, all right? I mean, life potentially been around for a couple billion years, but in terms of the most of life you're thinking of, it's in millions. And the rock layers that contain the fossils must be less than 10,000 years. Thus, all dating of all things, and whatever means that yield old age for things on the earth or rock layers are also falsified. You see how this one thing that he's come up with, this one idea, has just utterly destroyed all other ideas. Like, it's the total gotcha thing going on here. All canyons, glaciers, ice cores, varves must be less than 10,000 years. Also proves the Earth must be less than 10,000 years because the surface of the Earth is less than 10,000 years old, and there's no other catastrophe that could have produced all the fossils and rock layers except the worldwide flood. All right, so you get the idea. It's just going to go on and on and on here. A common descent of species is falsified as life has only existed for 10,000 years. That only leaves a common creation for the creator to explain all similarities, which is proved by the principle that all designed things have a designer. And since the Earth is less than 10,000 years, so is the moon. And that also means the long age dating for the moon is also proved false, right? So he has proved everything, false, by just looking at mutations. And I expect that's why he's sharing it onto many, 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 many groups, and with many, many, many different examples that are all the same example, just with different organisms, hoping that somebody's like favorite animal is a platypus, so they'll read that one. Somebody else loves a kangaroo, so they'll read that particular one. It is impossible for evolution to produce over 100 trillion unique genes, possibly over 500 trillion. All right, and these are just other arguments that are not really about the main thing we're talking about here. So where did they get the similarity percentage? Was it overstated? Right? Because that's his argument, is this must be overstated. It can't possibly be 50% similar, right? Because if they are, and he might actually agree that they are, we'll see, because they're commonly designed. If so, then all such similarity percentages are not reliable. Thus, there is no evolutionary descent. If too high, that proves the long dating of ages is too long. If very low, then there is no evolutionary descent. Thus, no DNA similarity can be attributed to evolution. If the percentage came out high, I'm still right. If the percentage came out low, I'd be right. In summary, all the time since the supposed last common ancestor branch off, the genomes of both would have been corrupted completely, and both lines would have long ago gone extinct. This falsifies evolution in billions of years completely. Any supposed DNA similarity would have been wiped out completely millions, eons ago. Let's see if we see any other. Let's see if he tries to explain what I'm going to say in a moment. Is he going to try to refute the objections to this? Does he know what the objections are? No new functional organisms, whatever. Even if we had trillions of years, we wouldn't be able to do that. This leads to the inescapable conclusion that DNA similarity between species that branched off from a last common ancestor supposedly millions of years ago can't be from a common descent at all, but proves recent creation from a common designer, common creator God. Same technique. Evolution in billion years are claimed to be a scientific theory, but as a scientific theory, it needs to stand falsification tests. But it's been falsified many times, so it's a false theory. Kangaroos are not your distant cousins, neither are all these other different organisms. My advice to all individuals, all schools, universities, organizations, and all governments should retract evolution in billions of years worldwide immediately. Evolution in billions of years should be renounced by all who believe it. All the time, the talent, the money wasted on false theories instead of being used to help people. Evolution in billions of years have brought untold misery and destruction to very many. It is one of the greatest delusions of all time and the greatest scientific delusion of all time. I think it's safe to say that I'm going to say that, this author is a tad bit delusional themselves. There may be real arguments and philosophical arguments and so forth, biblical arguments, about the age of the earth and so forth, which deserve some, some real discussion. And this might sound like an effective argument, but no. Let me try just giving you a couple ways to think about this. First one, I said fruit flies. Let's just start with fruit flies. Okay, so the world's 4,000 years old, right? This individual obviously believes the world is less than 6,000 years old, or 6,000 years old or less. And there were fruit flies in the original creation. And those fruit flies have been buzzing around, eating fruit and doing the only thing they really think about, which is reproducing, right? They try to reproduce within a day or two, of being born, right? And each fruit fly can lay hundreds of eggs. And each fruit fly has, several 100 million base pairs of DNA. I didn't look up fruit flies. I don't remember this. The thing is, you might think, well, it can't be the size of a human being, but there are tiny insects that have genomes that are the size of a human being. Genome size is highly variable. and there are some very seemingly simple organisms that have extremely large genomes. But let's just say they only have 100 million base pairs in their genome. And then they're going to reproduce, right? They have to produce a new generation of fruit flies. And they're going to do that, every couple days. Let's give it a week. And in tropical environments, that means they're doing it, 50 times a year. It's like you get 50 generations, right? 50 generations, and they're going to have, 30 or 40 mutations per generation, right? And then you start adding it up, and they have a fairly small genome. And as they add up, shouldn't they be experiencing incredible amounts of genetic entropy? Shouldn't their genomes just be chock full of mutations, even just over 4,000 years? Shouldn't they be on their last leg? Shouldn't they have disappeared already? And yet they seem fine, right? There's some really well-adapted fruit flies, and they don't seem to be suffering, all right, from genetic diseases and maladaptions. They're very fit for their particular environment. They're doing perfectly well. Add to that the fact that there are thousands of species of fruit flies, and potentially God only created maybe one of those, and it's become thousands of different species, each of which are reproducing and producing hundreds of thousands of individuals, which means there are billions and billions and trillions and quadrillions of mutations in the fruit fly population across the world every single year. Right? How did they continue their existence under this heavy load? right? So if you can explain why they're still alive, right, within the young Earth creationist timeline, then you can probably also explain why we're still alive, other animals are still alive, maybe after millions of years. All right, so we'll start there. And you just think about bacteria, think about other organisms that are like a bacteria that divides, E. coli every 12 minutes, right? And even after 4,000 years, if every 12 minutes it's dividing and making a generation, if you go back from the original ancestor of that bacteria to the one that's alive today, anyone that's alive today will have an ancestral lineage, right? Right. There's a lot of them and a lot of ancestral lineages, but you can just take one from today, one from the ancestor, and how many generations have there been? And they have 1 million base pair genome. I can tell you they've had more than 1 million mutations. More than 1 million mutations, which is exactly what his argument is here. He's saying there's been more mutations than the size of the genome, and therefore this organism should have fallen apart a long time ago. It shouldn't even exist. How can you completely obliterate your genome and still exist? So that you should look at that and go like, there must be another piece to this puzzle. It can't be as simple as the mutations add up and they break everything down and eventually there's nothing that works anymore because it completely changes every base. So with bacteria, I'm giving you the same scenario that this author is using to say he's completely destroyed billions of years. And I can say with bacteria, compared to bacteria today, all right, their descendants, it might be different species than they were originally. I have now just completely destroyed a 4,000 year chronology, right? Yeah, they can't be alive. They have to be dead for the same reason. Now, I don't think that's the case. I think those bacteria are perfectly fine. I think an E. coli today living in my stomach with the genome it has, all right, is very well adapted and does a really good job and is not in any danger of going extinct, right? Even though it may have quite a few different base pairs than it had 100,000 years ago or 1 million years ago or 10 million years ago or 100 million years ago. All right. I think E. coli, something like E. coli, been around for a long, long, long time. And it just keeps on trucking. And how does it do that? Because every generation, it makes mistakes. It replaces the base pairs with something else. Oh boy. So this is going a little longer than I had hoped. All right. I'm already sitting at 30 minutes. And I've really just introduced the problem. And I've given you some ideas about why it's not really a problem. I haven't given you an explanation yet. I've just given you some teasers to make you realize there must be a different solution. All right, there must be something else going on. All right, so let me try to explain this. Mitochondrial genome. All right, the mitochondrial DNA. That'll be another nice example. Right, so your mitochondrial DNA. Right, your mitochondrial DNA and mine, we each have a little genome in our mitochondria. It's about 16,500 base pairs in size. All right, tiny compared to our rest of our genome. And the mutation rate in our mitochondrial genome is actually, a good bit quicker than our nuclear genome. I'm gonna, every couple generations, you're passing a new mutation down in that genome, even though it's only 16,000 base pairs in size. So we have something called mitochondrial Eve, right? That's the original person, female, who had a mitochondrial genome. And they had one genome that had 16,500 base pairs of a certain sequence. And when they had offspring, female offspring, she gave those female offspring a copy of that genome. And it may or may not have made a mistake in that first generation. Let's say that one of them made a mistake, one didn't. So now the two offspring, one of them has a one base pair difference than the other. And then they have offspring, and then they have offspring, and they have offspring, and they have offspring, and so forth. And so she is the common ancestor between you out there, whoever you are, all right, whoever you are out there, and me. And there's a certain number of lineages, right? There's a certain number of ancestors that go from you back to common ancestor between you and me. And that's not necessarily the universal ancestor, because if you're somewhat closely related to me, well, then we share a common ancestor more recently in the past. But potentially we could be at the opposite ends of the human population, and we could trace all ourselves all the way back to that universal common ancestor of the mitochondrial genome. So there is a buildup of mutations in that genome. And what will happen though is, and what hasn't happened yet in humans, but will happen in the future, and this is where the millions of years comes in, is that initially you have there's a mutation rate. And so after every generation, we're substituting a new base, a new base of DNA gets replacing something else. And so the more generations that go by between you and me, I'm using this graph in a little different way than the axes have it, but there's going to be more and more and more mutations that are going to build up. All right. And it builds up at a fairly constant rate. There's sort of a substitution rate, the number of substitutions per generation that along each lineage. Right. So if you and I didn't share a common ancestor for thousands and thousands of years, then we would probably be up here somewhere. We'd have quite a few differences between us. If you and I shared a recent common ancestor, like within 3 generations, we might have identical mitochondrial genomes, right? And after 50 generations, we might differ by 3 or 4 base pairs. Now, it turns out, like all human beings, like even the most different one of us, only have about 100 differences. Right? 100 differences. That's not a lot. It's 100 out of 1,600. Right? So it's a nice little percent difference between us. What if the human race persists for longer and longer and longer periods of time? Well, you would expect what? You would predict that the number of mutations would just continue to rise, and they would go and go and go and go and go and go. And then what this author seems to think is, just at least in terms of the way they've written, is that after a certain amount of time, and you could calculate based on the number of changes per generation, you could just calculate out in the future. You go like, you know what? 65,000 years from now, or 100,000 years from now, there'll be 16,000 differences between some people on Earth. That's the entire genome. They would be 100% different in their mitochondrial genomes. How is that possible when the mitochondrial genome is super important and there are genes in that genome that are like essential? And how could I possibly change every sequence and still have those genes functioning the same way? I mean, could we really both be human beings at that point? Shouldn't we be something different? No, but that's not what's going to happen in the future. It really isn't. Because what's going to happen is, at first, every mutation that occurs is kind of a novel mutation. Like I had a mutation, somebody else has a different mutation, someone else has a different mutation at a different spot. But after there's a few mutations, guess what starts happening? Somebody else has, some other woman has an offspring. She makes a mutation in her mitochondrial DNA, and that mutation is at base pair position number 100. But somebody else already had a mutation at base pair number 100 from an A to a T, and you're making one that's an A to a T. That is a new mutation. It's a new mutation for that particular offspring compared to somebody else out there that has that. So it doesn't actually add new variation between those two people. In fact, it's a reversal. Two people can end up having the same base pair, even though they got it from 2 separate mutations. And so they're not actually getting more different at that location. How many times can you change the same location? There's only 4 base pairs. So you can change it to an A, to a C, to a G, to a T, but you could change it from an A to a C, back to an A, back to a T, but over to a T, then back to an A, and then to a G, and then to a T, and then to a C, and then back to an A. Right? There's only 4 different locations. This is saturation. Once the mutations have reached a certain point, a certain number in the genome, so there's this many variations in the population. Or if you just want to stick to 2 people, here's 2 people, and they're 10,000 generations separated from each other. And let's say they've reached a point where they're 15% different from each other. All right. Once you've reached 15, 20% different in the mitochondrial genome, At that point, you start to have the same mutations reoccurring over and over and over and over again, rather than other mutations that didn't exist before. And therefore, the rate at which you're accumulating new mutations slows down, right? It can't keep going up because you're constantly creating some of the mutations are repeats. And so you get this sloping off instead of this straight linear progression. And eventually this is going to peak, right? And it's going to peak because at some point you're always making mutations that remake the original genome, right? They go back to the original state, right? So this is going to peak, but it's going to peak for another reason as well, right? Here's the other thing that's happening. All right. Let me continue with that thought for a second here. Let's think about the whole genome. How different could it possibly be? Really, 25%, right? It's not zero. It's never going to reach 0% similarity where every single base pair is different. That can never happen when you just have mutations occurring because of these back mutations. So I did a really quick experiment that any one of you can do to verify this for yourself. All right. Go to Claude, Gemini, ChatGPT, and just say this. Just say, generate 2 DNA sequences with A, T, C's, and G's randomly, right? They're going to create 2 random 500 base pair sequences. Then align those 2 sequences from the first base to the last base. So it is randomly generated 500 base pairs, and then it just lines the 2 up, first base to the 500 base. And then calculate the similarity between those 2 sequences. And can you predict how similar they'll be? In other words, this is random sequence, right? You're generating random sequence and you're just lining it up. And I ask you, what will be the percent similarity between those 2? You should be able to predict what that is. This is my class. Let's say, all right, let's take a minute, pause, do the calculation, and then we'll give you the answer. But here's the answer. All right, 25%. 25%. I did it. And out of my first try, getting ChatGPT to do this, it gave me 24.7%, which was well within the statistical bounds, right, of expectations, because there's going to be some randomness to there could be accidental, right? By chance, you could end up with a few more sites that were similar than you expected. But you expected 25%. Why? Because at every position, you can have either an A, a C, or a T, or a G. To have every position be different and be 0% the same, it means base pair position 1 has to have an A and the other one has to have something else. And then you go to base pair position 2, you have to have a base pair, like let's say it has a C. Well, then the other randomly generated strand has to have something else, one of the other 3. 25% of the time, if you had an A there, and then you randomly generate the other one, 25% of the time you're going to generate an A, in which case you're going to have the same base pair. That's going to be similar. There's your similarity. And so 25% of the time, at 25% of the locations, should end up having the same base pair as the other strand, even though it was randomly generated. So 25% sequence similarity. So if you took sequence from 2 different organisms, all right, and you just sort of lined it up. Let's say you had 2 genes where you knew where those genes were and you could kind of line them up, and there's some sequence in between, and you can't really I can't line it up because I can see the similarities all. You just say I just line the sequence up. It should be about 25% similar, right, by chance. So you're not really ever going to drop below 25% similarity. And the 30% similarity thing is kind of ridiculous too, for this reason. Organisms have similar genes, right? Why don't mitochondrial genomes ever get more than, 25%, 25 or 30% dissimilar? In other words, they're still 75% similar to each other, even though they're in totally different types of organisms. That's because they're about the same size. They contain the same kinds of genes, and some of those genes are really conserved. A thing that was never mentioned in this person's dialogue, they never mentioned natural selection. They never mentioned the force of mutations happen, and mutations happen randomly, but the substitution rate is how many survive to the next generation, right? If you have, what I'm showing you here is, this is an alignment between two sequences. So if you had two sequences in a genome, and here's some genes, and all these little blue and red bars represent conserved elements with important functions. So like they're, if you don't know, you're thinking like protein coding genes, right? The genes that make like insulin, right? That's a fairly conserved gene. You and I have almost the same sequence in that gene. But if you were to go to other parts of our genome that don't make an important gene, you and I would have more differences. Why? Because when we have mutations there, we go like, well, I don't care. They didn't really change me, right? So they randomly accumulate over time. And the mutations can change from an A to a C to a G to a T, back to an A, back to a C, back to, and over time, if we're, have enough generations, eventually those those sequences, those areas will only be 25% similar to each other, right? They'll lose any similarity. Your sequence and my sequence in our descendants, eventually there'll be areas of our genomes that won't be more than 25% similar, or be around 25% similar. But there'll be many, many places in our genome, and in our mitochondrial genome especially, because it's super important and all the genes are compact in there. There's not much space between the genes like there is in this particular example here. So like here's a really important gene, and between two fairly different organisms, I can't remember what the two organisms were in this particular example from my text. The sequences are 75-80% similar. I remember these are two really divergent organisms, very different types of organisms. Think of like, I don't remember the exact ones, but we'll say like a fish and a human being, right? And so they're quite similar where the genes are that we have in common, because you and I share a large number of genes with other mammals, especially, but even fish. I have a leptin gene. Fish has a leptin gene. I can line up a leptin gene from a fish with my leptin gene, and a dog's leptin gene, and a salamander's leptin gene. And I can see that a dog's leptin gene is more similar to me than a fish is, but nonetheless, I can clearly see a fish is 65-70% similar. The dog is 95% similar to me. But anyway, in these important places in the genome, we have high similarity. In places that are between genes and we can't find any function for them, they're just, some sequence that's in the space between these genes. What we see is they don't have even 50% similarity. They're below 50%. I said, you could have put a 25% line here. They really wouldn't go below 25%, because theoretically you can't get more than 25. You can't get less than 25% similarity, because just by chance, you're going to keep generating similar sequences by accident in 25% of the locations. So what you'll find is that if you lined up me and you and I, we would be 99% similar across here. We'd be 100% similar at most of our genes. Like most of the coding sequence of our DNA is 100% similar. Between our genes, we don't really care so much about mutations, and so we might be 98% similar in some locations, or 97% similar in some locations. And so you would get this kind of map for us too. So what does that mean with respect to our little Facebook thing here? When you talk about being 50% similar, yes, you certainly do expect to have high similarity among some portion of the genome. He's talking about across the entire genome, saying like 50% of the entire genome. There's huge other complications. I'm sure. Yeah, I don't have time to go into all of them. Let me give you the quick snapshot of them. One would be our genomes aren't the same size. So how are you going to count similarity? If my genome is half the size of a plant, does that mean we're 50% similar because it has twice as much DNA as I do? And then what about the sequence we have that is the same genes? Well, then we might be 50% similar for those. How do you calculate similarity for the whole thing? Are you only calculating the similarity across sequences you can actually like line up? I mean, you might be missing 1 million base pairs in your genome compared to me, or I might have 1 million extra base pairs. This often happens. We have chunks where we've copied our DNA or lost pieces of our genome, in which case you and I really aren't. We're 99 point something percent similar in all the sequence you can line up together. But then how do you count the chunks that are missing? That's like 1 million base pairs that are missing. Should I count that as 1 million differences between you and I? In which case we're not actually that similar. Right? So it is actually not a trivial thing to produce a similarity matrix. You have to define what that is. This person doesn't define what that similarity means. And I'm not sure he understands when other people say that a banana is 50% similar to human. I think they're really just talking about the genes that are there. How similar are those gene sequences that we have in common? Not talk about all the other space, because all the space between the genes is going to be 25% similar. Right. So what I'm really interested in anytime I would look at two different organisms, I'd want to know how similar are their conserved genes or the genes that we have in common that we're doing common functions with. That what I expect to be high similarity. I expect other areas to be low similarity. But now here is the main rub. This particular author hasn't spoken about what natural selection is doing. Right? There's going to be mutations that occur. And just because there's 4.5 billion mutations over, 100 million years, doesn't mean those 4.5 billion mutations are going to are going to replace every piece of DNA. They're not, if this was, again, this is the insulin gene, or let's say this is ATP synthase. That is the gene that puts together adenine triphosphates, which are your energy carriers. And without them, you'd be dead, right? And that's all animals, right? All organisms make ATPs. So if you make mistakes in that gene, you're dead. So mutations do happen in those genes, but then the organism dies, in which case they're not part of your lineage, right? I mean, that lineage doesn't exist anymore, but other members of the species don't have that mutation and they persist. So those mutations happen, but they never persist. So the substitution rate in this particular area is extremely low. Substitution meaning you actually substitute it and it survived and continued to the next generation. The substitution rate is nil in some areas of your genome, which is why your ATPase, my ATPase is probably almost identical, except at a few locations where it doesn't matter what the base pair is there. And your ATPase is very similar to a dog's or fish, or, we have high similarity between some sequences. So some genes have nearly the same sequence in very different organisms because every mutation that happens to them is lethal, or at least highly selected against over time, over many generations, so eventually disappears. And so what you end up with is all the mutations all the 60 or so that you're giving to the next generation, guess what most of them are? They're mostly in this region in here. And because they're in this region here, which isn't doing anything, you're just changing an A to a C to a T to a G, back to an A, back to a T, back to a C, back to an A, back to a G, back to a T, back to an A, over and over and over and over again over many different generations. So therefore, yes, you can accumulate in a lineage of any lineage you look at. If you say but this lineage has 75 million generations. Yes, it's true. That lineage will have experienced 4 billion mutations. But it doesn't mean their whole genome is going to be obliterated. It means that some of these spaces will have changed over and 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 over again from from an A, again, I'm going to say from an A to a C, back to an A, back to a C, back to an A, back to a C, back to an A. Right? Is the genome really getting that different? Especially if it's in a region where it's I don't care what bases I have there. I really don't. They don't really matter. In which case the organism isn't changing as much as you think. right? It's only going to be changes that occur in genes and regulatory elements, things that control genes. And those sequences also we see don't change over time very much. And again, they don't change not because they don't experience mutations. They absolutely do. It's just that if there's a mutation to an important regulatory gene, that individual is not going to survive well, in which case they don't have offspring. And it's only other individuals that have mutations in the spaces that survive. They go on and have more offspring, and they become the actual lineage that survives. This would be one of the primary reasons why you can experience enormous numbers of mutations in your deep lineage, or you can also experience enormous number of mutations in you as an individual, because most of them are happening between your genes. And any of them that occur in important genes are going to kill that cell. But you have lots of cells in your tissues. And so you're like, whatever, I lost that cell, right? That cell died because it had a mutation in ATP synthase. I couldn't make enough energy in that cell. The cell died. I have lots of other cells to take its place. They then are the the progenitors of the next generation of cells in that tissue that don't have that mutation in that gene. But they will make mutations in other places. But as long as they're not important places, they will accumulate mutations in non-essential regions. Now, unfortunately for us at my age, maybe your age too, you really have accumulated some mutations in genes that are doing functions that are important to you. And they are gradually decaying you. And eventually, you as an organism is going to suffer as a result of those trillions and quadrillions of mutations. But your sex cells are protected from a lot of those, and you're not going to pass many to the next generation. It's one of the reasons why all organisms reproduce rather than just live like thousands of years, right? No individual can survive that kind of damage, but by passing it to the next generation and allowing natural selection to select for individuals that don't have lethal mutations or really bad mutations, you go on to form new generations. I was going to say a bunch more stuff. It's like there's 4 or 5 more reasons why mutations don't accumulate and how we get rid of mutations. After, some types of mutations which are bad, we can actually sweep out and get rid of in our populations, not as individuals, but as a population over time, we sweep out. And so this isn't the gotcha that I think this person thinks it is, right? They think that this simple math is disproving the fact that an organism can survive for millions of years as a lineage. But the answer to that is quite simple. If you kind of boil what I've said here down, the numbers don't add up the same way he thinks that they add up. I'll leave it there. Let's stop because I've hit the 1 hour mark, and I said to myself, going over an hour today on this topic. Not quite worth it. But if you don't know molecular biology and genomes and stuff like that, I hope that this is at least interesting. And I also just made it because I want to be able to point to this whenever somebody shares this particular Facebook proof that mutations should be killing people after thousands of generations, when this just really isn't the case. And we know it for some of the reasons I gave you here, plus I promise there are many other reasons that we could go into. All right, that's it. Thanks for listening. Subscribe, like, all that other good stuff. And we'll talk to you later. Bye-bye.